Hi, and good evening, everyone. My name is Joy Bowie, and I have the pleasure of serving as the Vice President of Research for the Lupus Foundation of America. We are so delighted to have each of you join us this evening as we talk about breaking barriers in lupus treatment, unlocking the potential of CAR cell therapy, T-cell engagers, and beyond. We have an all-star lineup this evening for you guys, dynamic speakers who are well-versed in these topic areas. But more importantly, we have a person living with lupus who is going to share their story this evening. So we are very excited to present this very important information to all of you. But before we get started, I want to go over a few housekeeping items for this evening. So the first thing, if you are experiencing any technical issues, please email Safwa Adjemang at adjemang@lupus.org. You can go ahead and take your phones out and take a picture of that, or you can take a screenshot so that you have that information close by, just in case you get disconnected. If you do, by chance, get disconnected because of technical issues, or you lose service with your wireless, our internet service, go ahead and just use that link that we provided you when you registered to log right back in. And if you can't, please reach out to Safwa Adjemang and she'd be more than happy to help you. Do want to talk about a little bit of question and answer etiquette this evening. So questions can be submitted at any time using the Q&A function at the bottom of your screens. We will not have the chat live this evening, but again, you can submit any questions that you have using that Q&A function. We will do our best to answer your questions, and I've already asked the panelists to keep their eyes on the Q&A box so that they can respond to your questions as they're coming through during various presentations. Please make sure that you keep your questions respectful and certainly relevant to the discussion this evening. We want to go ahead and share that we will have a post-webinar survey that we'll be disseminating and sharing at the end of this webinar. And that particular survey, your responses will help us improve our research program. So it's imperative that you participate so that we can make sure that we're delivering information, content that meets your needs, and certainly we have the opportunity to respond to unmet needs as well. We also want to let you know that there's an opportunity to sign up for Research Accelerated by You, which is our lupus patient registry. And so if you haven't heard about RAY and would like more information, please indicate that when you're filling out the survey. Lastly, after the webinar, we will send a follow-up email with key takeaways, resources, and any relevant links, including a link to this recorded webinar that will be posted on our YouTube channel following the presentation this evening. Next slide, please. So we are so privileged to have wonderful academic researchers with us this evening who have spent their careers studying lupus, treating lupus, and really raising awareness about this disease. We also have a person on our panel who has lived with lupus for quite some time now and has had, the opportunity to experience CAR T-cell therapy as part of her current treatment regimen. And so we're going to be listening to presentations and talks from all of these individuals, and we're so thankful that they're able to join us on this evening. Next slide, please. We also want to thank our sponsors who have been more than supportive of the work that we do here at the Foundation. We're very grateful for their support, and without them, these types of programs would not be possible. So thank you to BMS, Cabaletta Bio, Century Therapeutics, Cullinan Therapeutics, Fate Therapeutics, Nkarta, and Novartis. Next slide, please. So this evening, we really want to set the stage by talking about clinical trials. Right now, we know that there are an abundance of clinical trials that are currently in the pipeline within the US and beyond. The last time I checked, I saw on clinicaltrials.gov that there were over 140 clinical trials that were actively recruiting people with lupus. And so oftentimes we'll have these types of presentations without first level setting about what a clinical trial actually is. So as this is an encore presentation, I wanted to give Dr. Anka Askanase, who is a professor of medicine and director of the Lupus Center at Columbia University, the opportunity to tell us about clinical trials and why it's important to participate in clinical trials. Dr. Askanase, thank you for joining us this evening.
It is a privilege to be here and talk about clinical trials and learn about CAR T cell therapy and the promise of CAR T in lupus, and share what we've learned with patients, people living with lupus, and hopefully helping them make these decisions whether CAR T therapy is right for them. So I'm going to try to do Clinical Trials 101 and why it is important to participate. So what are clinical trials? What are the phases of the clinical trials? The role of the informed consent and patients' rights, and how those protect patients from any infringement on their rights, and then how clinical trials continue to treatment as advancements. The slides that I'm going to share with you were developed as part of our Department of Health and Human Services Office of Minority Health grant award program, and we had put them together, and I hope that you'll all appreciate the work that went into them from patients and colleague feedback, and a designer helping us with getting those organized and ready to share them with patients and our patients in the clinic and with you all. So next slide, please. So what is clinical research? Clinical research is medical research that involves people. It led to the discovery of every disease treatment prescribed today. Clinical research might be right for you if you want to help others, receive regular monitoring, or test new disease treatment that might work better than those currently available. Next slide, please. In terms of the types of clinical research, there is observational research, where we monitor people over time, and interventional research, the clinical trials, where we test a drug, a device, or a procedure. Next slide. So what is a clinical trial? Clinical trials are research studies involving human volunteers to evaluate medical products like medications, vaccines, or devices for their safety and effectiveness. So if they are ready to be released for the use of people everywhere. To continue developing new treatments, lupus clinical trials need diverse participants representing the populations most affected by lupus. And the Office of Minority Health launched a campaign called Let's Take Charge to help spread awareness of clinical trials for people with lupus and increase their participation in clinical trials. The hope is that clinical trials for people with lupus will become part of their clinical care, that this is going to be an integrated care where, much like for some cancers or rare diseases, clinical trials are part of the way doctors take care of their patients. Next slide. So we're going to do a little bit about the 5W, what, when, where, who and how. So what can you do? Well, you need to learn more about clinical trials, and there you are learning, and then participate in clinical trials. Next slide. Who participates in clinical trials? Well, everybody should participate in clinical trials. We just, I just made the point that clinical trials should be part of clinical care. However, different studies have requirements, either the medical conditions, the treatment history, the disease type and stage. Others include people of different races or ethnicities, gender, age requirements. So all of these need to be taken into account when the physician and the patient make these decisions. Next slide. It ultimately comes down to a pros and cons weighing, understanding the benefits and risks of participating in clinical trials. And while here it might look like there are more benefits than the risks, I'm trying to keep the balance even so that it's very clear that that balance is different for every individual. Ergo, these are individualized decisions. Some of the pros, you help other lupus patients get better treatments in the future. You have early access to treatments. You can learn more about your health. Expenses are covered in clinical trials. Medical care is covered by the trial, so there's no expense to the participant. You may get more information, supports for lupus patients. You'll get more frequent checkups. You have increased access to the study staff, and sometimes you can get paid. The cons are, well, you sometimes don't get the drug. You sometimes get the placebo, the sugar pill, or the sugar infusion. Well, it's sugar. It's not sugar. It's an inert substance that does not have any effect on your health. It may take more time. There are maybe side effects. You're going to need more blood tests. So all of these need to be weighed carefully before making that decision. So your doctor and you will think about this and make decisions. Next slide. So what is needed from you? Clinical trials have their own set of rules known as the study protocol, which includes the testing schedule, the procedures, the dose to be administered, the medication, the frequency, and the things that are going to be happening within that doctor's appointment. And sometimes these studies can be long, a year, 2 years, and the new cell therapy trials are now asking us to follow patients for up to 15 years, a long time. Next slide. So a little more. How, when, where do you find information on clinical trials? First, ask your doctor. How do I decide what trial is right for me? Ask the questions. And to learn more, we suggest that these are trusted sites that will provide you more information about lupus clinical trials, clinicaltrials.gov, and the Let's Take Charge site from the Office of Minority Health from the Department of Health and Human Services. Next slide. So what should I do? Well, what should you do if you're asked to participate in a clinical trials? Well, first you can say yes, but of course you can say no. You can ask as many questions as you need to understand the trials, the risk, and the benefits. Ask for the contact information for the research staff in case you want more information, you want to think about it, you want to talk with your family and your friends, and then come back and either decide to be a part of this study or say no. Next slide, please. Why is participation in lupus clinical trials so important? Lupus is complex. Each patient is different. Lupus clinical trials are difficult. There are not enough patients for all studies. We need a lot of people in order to study the new drugs and bring new medications to people with lupus. Next slide. So Your participation in clinical trials is the reason why multiple new medications have been and can be approved and available to all people with lupus. And ultimately, we hope lupus will have a cure. So we suggest that you too should be part of the breakthrough. Next slide. This is a really beautiful slide that I borrowed from Joy, that has put it together for the LFA. So what are the phases of clinical trials? So what is the process to get from the laboratory to bringing the drug to the patients? First, preclinical, so before we test it in the clinic. Laboratory studies, sometimes it takes a long time to get these medications that seem to make sense to test in patients, in healthy volunteers, in people with lupus. The phase 1 is about safety. It takes several months, evaluates safety, and gather information about how that drug interacts with a human body. If if the drug is safe and it looks promising, the next step is a phase 2 that evaluates safety and dosing. Again, it takes about several months. Further value on safety, monitoring side effects, decide on the best dose, and check if start thinking about, is this drug effective? Is this treating the problem? And then phase 3. This is safety and efficacy. Sometimes this takes years because doing studies on a large number of patients is difficult. So you see the number of people on each of these steps gets bigger and bigger. Phase 3 clinical trials are much bigger. In lupus, there are several hundred patients. The first studies that led to the approval of belimumab actually included 2 studies of 800 patients each. 1,600 patients were part of that particular drug development process for the phase 3. And then phase 4, the drug is approved. We have achieved this big success where the drug is approved by the Food and Drug Administration and it's released for use in patients. But sometimes the FDA says we need more information about efficacy, effect in various populations. Belimumab had a study that was mandated to include only patients of Black race so that we better understand how it works in people of Black race, and then evaluate side effects. Is there an increased risk for infections? Are there cardiovascular side effects? So all of this information is gathered in phase 4 studies. Next slide. So what happens in the clinical trial? Always, clinical trials are optional. Yes, we advocate for them. We would like people to participate, but it is ultimately your choice. Sometimes clinical trials take place in the hospital, in the doctor's offices, in the clinics, or in university practices. Now, more and more, there's interest in doing trials that are decentralized or partially decentralized. That means that a lot of the trial activities and a lot of the evaluations can happen over telemedicine from your home. And all of this is evolving, and lupus trials are probably going to also become more decentralized, where some of the evaluations are going to happen over telemedicine or with devices that you'll get to wear, and then you don't need to come to the doctor's office and travel and spend time on the road. A new medication is tested. The new medication may be different from or in addition to the regular medical care you receive. A lot of the lupus trials are in addition to your regular medication, which we call standard of care. We have to study safety, how safe is the medication, side effects, problems that occur when treatment affects healthy organs, efficacy, how does a treatment work for the health issue that it's supposed to treat. Next slide. We already went over the phases of the trials. Next slide. I put here more details. And this is a little more detail about the success story of the development of belimumab or Benlysta. And you'll see that it took about 10 years to get from the phase 1 to bringing those medications to the patients living, the people living with lupus. Next slide. So a little bit, couple of words about how are patients' rights protected in clinical trials. Next slide. A lot of my patients want to make sure that they are still being treated and their drugs that help them be in a good enough place are not taken away. So standard of care is important. A lot of the lupus trials require patients remain on the standard of care, but you learn in a little bit that some of these cell therapy studies are saying the drug is powerful enough that we think we're not going to need that. The standard of care is the treatment that is accepted by medical treatments as the right treatment for the disease. So a little more about what happens in the cell therapy trials and how we're addressing removing the standard of care. Next slide, please. So patient safety is top priority. There are laws in place to protect people who participate in research. The FDA regulations protect safety and privacy of the participants, and there are two major ways that protect patients. The first one is informed consent, and then the institutional review boards that at the level of each practice, each institution are there to make sure that your rights are protected and you are not subjected to anything that can cause harm. Next slide. So the informed consent is the process of giving potential participants the facts about the trial, to discuss safety, to discuss the balance of risk and benefits, to discuss your rights. This informed consent needs to be the first step before there is anything that has to do with a trial. Next step. So before participating in the trial, it is not a contract. You're not giving up your rights. This is about understanding the purpose of the study, understanding the risk and the benefits, and make sure that this is right for you. Next slide. The research team includes investigators, research nurses, research coordinators, and data managers, all there to make sure that the trial is conducted safely and according to the rules put in place by the Food and Drug Administration and by the institutional review boards. Next slide, please. And there's a lot of important protections for your confidentiality to make sure that your personal and medical information are kept confidential. The members of the research team, people monitoring the study, people from the FDA can have access to your medical records, but none of the data is released to the rest of the world. None of your personal data is used in any publications or in any communication that is spread widely. Next slide, please. So this is our funding that allowed us to put together these slides. And I just want to mention that as the world evolves, clinical trials for lupus with cell therapy are probably not going to follow the same pattern of phase 1, 2, 3, and 4. It seems that the Food and Drug Administration suggested that data from phase 2 trials may be enough to get the drugs approved. Additionally, they also talked to researchers in the cell therapy field about the possibility of having everybody be on the active drug and a single arm without that placebo arm that allows for comparison. The reason for that is that the patients in these trials are patients that have a serious condition, and that the efficacy from these drugs is expected to far exceed that from other normal medications. And with all that, I hope that I've made a good argument that clinical trials should be part of standard of care. And I'm going to pass the baton to my colleagues to tell you about cell therapy.
Thank you so much, Dr. Askanase, for that very informative presentation on clinical trials. So now we really understand what it means to participate in a clinical trial, while they're so very important, and also why it takes so long for a drug to be approved, right? It's a very long process. So next we have Dr. Elena Maserati, who is the Vice Chair of Clinical Affairs in the Department of Medicine at Brigham and Women's Hospital. She's also the clinical director of the BWH Lupus Center, director of clinical trials at the Lupus Center as well, and she's an associate professor of medicine at Harvard Medical School. She's going to tell us a little bit about B cells and why they are so important in lupus, and why targeting a B cell might be a promising approach to treating the disease. Dr. Maserati, thank you for joining us this evening.
Good evening, everyone, and it's nice to be with you. I hope that this webinar is informative to you. And aside from this, feel free to reach out to me by email at the Brigham, and that's at emazerati@bwh.harvard.edu. So thank you. Next slide, please. These are my disclosures. Next slide, please. So just some broad points to begin with as we get into a little bit of biology in a few minutes. So as many of you know, lupus can affect many organs. The most common organs are the skin and the musculoskeletal system. The musculoskeletal system being joints, muscles, etc., but especially joints. So skin and joints are the most common organ systems, but many organs can be involved in the manifestations of lupus. Kidney disease is seen in about 40% of patients with lupus. There can be cardiac involvement and lung involvement. And so nearly every organ in the body can be affected by lupus, but usually not simultaneously. And the process by which the disease manifests in an organ can be triggered by different pathways, by different immunological or biological pathways. So that's to say the same processes might not necessarily result in the same clinical manifestation in a patient, but there are some common characteristics. So regardless of these, whatever clinical manifestation of lupus a patient may have, whether the patient may have, let's say, joint disease or heart disease or primarily skin disease or a combination of all three, the principles of treatment are essentially the same. Next slide, please. So the guiding principles, there are two major guiding principles. One is to really control the inflammation of the organ that's being affected as early as possible. And the second is to minimize or eliminate steroids to the extent possible. We know that corticosteroids, medications like prednisone and methylprednisolone, work to control disease activity. And I tell many of my patients, gee, if steroids weren't associated with side effects, no one would have to probably see a rheumatologist. We just give everyone steroids. But the problem with steroids is, though they control inflammation in many different organ systems, they have unacceptable long-term side effects. And that's a major reason why whatever treatment we decide to use and whatever organ is affected in the particular patient, that we eliminate steroids. Next slide, please. So what causes lupus? Well, we don't know the cause of lupus. We have some good ideas about triggers, but one of the major challenges in lupus is finding out the cause. And we know that it affects the immune system, that it's what we call the prototypic autoimmune disease. And autoimmune diseases are characterized by the formation of antibodies, which are produced by B cells. And antibodies, autoantibodies, in lupus, we have autoantibodies, namely antibodies that attack organs in the patient's body. So we know that antibodies are produced when a patient is exposed to infection. So, for example, with the COVID crisis a few years ago, we produced antibodies to surface structures of the virus that was responsible for COVID, and we generated antibodies to them. In a disease like lupus, where autoantibodies are formed, the patient's B cells produce antibodies that react against structures within organs of the patient. So there's dysregulation of the immune system. And what does that mean? Well, we all probably produce antibodies, autoantibodies, a low level of antibodies, but in a disease like lupus or in lupus, the control, the regulation of the production of antibodies goes haywire, and numerous autoantibodies are formed for the patient. So there's what we call aberrant immune cell activation, and different components of the immune system are involved in the development of lupus. So the immune system involves cellular components like T cells and B cells, which fall into the category of lymphocytes. And then there are other immune cells, other cells that participate in the immune system of the body, neutrophils, monocytes, and other cells. And then there are molecules in the circulation that are produced by these cells that also regulate inflammation. And an example of those molecules are called cytokines. Okay. So there's immune dysregulation, there's disruption of that immune system. And then in some patients, there's a genetic predisposition. So when we meet patients, we often ask them if there's any autoimmune disease in the family, because it's not uncommon for patients to have had a parent or cousin or other relative who's had an autoimmune disease that might share some features of lupus. And there are then there are modifications to the gene that result in the development of lupus in a particular person. It's not that there are environmental factors that lead to lupus. We ask patients to avoid ultraviolet light because we know that sunlight can trigger the disease. So all of these factors are thought to participate in the pathogenesis, in the cause of lupus, but the specific cause, the specific trigger, is still not known. Next slide. So how can we make use of our knowledge of the immune system in helping to fight lupus? And one of the things that's happened in medicine in the last 25, 30 years is a clearer understanding of the immune system and technological advances in identifying cells and mechanisms by which these cells work in the development of lupus. And this is a little bit of a busy slide, but what I'll share with you are some of the cellular knowledge that we've learned about the cells involved in the pathogenesis or in the cause of lupus. So on the left-hand side of the slide, you'll see a B cell and a T cell, and then you'll see a dendritic cell, and you'll see that there are molecules that attach that allow these cells to communicate with one another. And it's a fairly busy slide, but the point being that the B cell is the major producer of antibodies, and B cells again are involved in producing antibodies for when we fight infection, and also produce antibodies that attack self in patients with an autoimmune disease like lupus. For the B cell to produce an antibody, it needs to communicate with other cells in the immune system, the T cell and other cells as well. And so then there's generation of autoantibodies. The antibodies are reflected here, and then the antibodies form immune complexes, which the antibody recognizes an antigen, which is what it attacks, and then the complexes trigger an inflammatory process where these molecules are produced called cytokines. So the B cell is stimulated to produce antibodies by communicating with other immune cells. Antibodies are produced that recognize antigens. The antigens can be external, like in virus, or they can be internal, like structures within the human body. And then the interaction of the antibody with that antigen results in the production of molecules that lead to the inflammation in the patient. In the case of the kidney, you have antibodies that react against components of the kidney that results in inflammation within the kidney. So that's fairly simplistic, but that's kind of like generally what happens. So as we've learned more about the immune system over the last 25, 30, 40 years, we have the opportunity to target specific components of the immune system to disrupt that inflammation that occurs on that right-hand side of the slide. So how can we disrupt things up front so that we don't produce antibodies that result in the recognition of self and then the inflammation that results and then the destruction or damage, if you will. Remember, our goals are to control inflammation early, to get at this process on the right side of the slide earlier and as quickly as possible. But how do we do that in a way that leaves the immune system intact enough to do what we want it to do, to fight infection as well? That's the challenge. Next slide, please. So we look at targets for intervention. So one of the things we can do is what we call diminish antigen exposure. Stay out of the sun. Avoid drugs that we know can trigger lupus. Reduce antigen recognition and T cell activation. So try to minimize that interaction of that antibody with that antigen. And we can do that with medications that interrupt that communication. We can get right at the B cell, prevent that B cell from making antibodies by either killing the B cell or reducing its ability to produce antibodies. And then the other thing we can do is interfere with the inflammation that results from the interaction of those antigens and antibodies up front. Next slide, please. So I just want to kind of get back to treatment that Dr. Askanaz had kind of alluded to a bit. So as a physician, when we sit and meet with patients and discuss treatments, we use information to help decide upon what medication, what treatment is best for that patient. And how do we decide on the treatment regimen? Well, there's anecdotal data. So, well, I tried this drug in this patient, or I used this dose and that seemed to work. So that's trial and error. And then we use the literature, and sometimes we publish studies of patients involved in or what we call epidemiological studies or observational studies that give us some information about what happens in a particular population. But clinical trials remain the most effective way to test whether a drug works or doesn't work, because it's somewhat of a controlled environment, and it allows for us to bring patients in who have very similar characteristics and then test the drug in one group and not in the other. And so what we do in clinical practice is we try to identify which organ system is involved in the patient and then pick the appropriate treatment based on these avenues of information, anecdotal data, published literature, but really clinical trials is the most solid piece of information that we have available to us to determine whether a drug will be helpful. Next slide, please. So this is just a little cartoon that I've made up. When I look at a trial, I think of three things, basically. So the population of the patients, who are the group of the patients being studied? So Dr. Askanaz mentioned the belimumab trials, the Benlysta trials, that belimumab was approved in 2011. And so that those were patients, those groups of patients were patients who had lupus, were failing standard of care, and had various manifestations, but could not have kidney disease and did not have any brain disease related to lupus. And so belimumab was the drug. The population was that population of patients. And then there was a way you have to measure what the efficacy of that drug is or isn't. So what did they actually look at? And in the lupus trials, they looked at what's called a composite score of disease activity. Next slide. So I wanted to kind of bring this up because I think one of the big success stories in rheumatology has been rheumatoid arthritis. And in rheumatoid arthritis, autoantibodies are also formed. B cells form rheumatoid factor. But we've been much more successful in treating rheumatoid arthritis than in lupus. And why is that? Well, the diseases are really different from one another. Rheumatoid arthritis is much more homogeneous, and as I've said, and Dr. Ashkenazi said, lupus is much more heterogeneous. So there are patients who have heart disease and joint disease, and then, in the next room when I'm seeing someone else, they might just have skin disease and joint disease. The diagnosis of RA is a bit more uniform than in rheumatoid arthritis. In RA, we had clear guidelines. We have new guidelines for lupus nephritis and lupus. Rheumatoid arthritis is a much more common disease. Lupus is a much less common disease, and our trial outcome measures. We still haven't arrived as a community in defining a consistent set of outcome endpoints for clinical trials next study. And this is going to get into B cells in a moment. Again, next slide, please. So these are all the drugs on the left that have been approved for rheumatoid arthritis, and then the right for lupus. And so belimumab was approved in 2011, and anifrolumab about 3 years ago, voclosporin right around the same time. But numerous trials have been undertaken, and I think this reflects how challenging it is to really understand what the mechanism of disease is in lupus. It's a bit clearer in rheumatoid arthritis. Next slide, please. So the treatments that have evolved, belimumab, anifrolumab, voclosporin, attack different components of the immune system. And this slide shows how drugs that have been tried in lupus in the last 20 years or so, and current drugs approved for lupus, affect different components of the immune system, especially the B cell. So the B cell here is on the bottom left, and you'll see that there are these little structures on the surface of the B cell, and these are receptors. And different B cells are characterized by different receptors. And there are antibodies that are directed against various receptors on the B cells that can serve as a way to minimize autoantibody production in a patient with lupus. So an example, some of you may know about rituximab. So rituximab is an antibody, a manufactured antibody that attacks this receptor, the CD20 receptor on the B cell. A number of years ago, there was another study of another antibody directed against B cells that failed clinical trials called epertuzumab, and it affected this structure, CD22, which is different from CD20. A new drug affecting this receptor, the CD19, at the top, at the 11 o'clock, if you will, on the B cell, was just recently approved for another rheumatic disease, and that is another cell surface receptor. So that's on the B cell. And then there are also receptors on the T cells above the B cell that you'll see that can be potential targets in the treatment of lupus. B cells result in the production of plasma cells, and again, plasma cells related to autoantibody result in autoantibody formation. Again, an antibody joins with an antigen, and then you develop this immune complex that results in inflammation of a particular structure. In this cartoon, you see the B cells activated, plasma cells are formed, create antibodies, autoantibodies, that then hook up with an antigen to form immune complexes and affect a structure within the kidney called the glomerulus. Next slide, please. So these are all the drugs that failed to meet endpoints that targeted different components of the immune system, various components in the B cell or in that communication pathway between B cells and T cells or dendritic cells. Next slide, please. So here are the B cell targets. So the B cell is one of the important targets one of the important cells in the development of lupus. And B cells are formed in the bone marrow and then result in antibody formation that goes into the circulation. And on the left side of this panel, you'll see the different receptors that are located on the B cell. And as the B cell matures, different receptors are located on each of the B cells. And we're learning more and more about which B cells are critical in the development of lupus. So as I said, rituximab, which is the second, attacks the second molecule here, the CD20, is found in pre-B cells and memory B cells down here, but it's not found in these pro-B cells. Whereas the CD19 antibody that I mentioned just a few minutes ago does affect this B cell. So what we're learning is, which B cells are most involved in the pathogenesis of lupus. But rituximab, which has been an effective treatment for some manifestations of lupus, depletes these B cells. So what we've learned is that depleting these B cells, diminishing the number of B cells in the tissues affected by lupus or in the bloodstream, leads to improvement in some but not all clinical manifestations. So there was two studies done around 2005 that looked at rituximab for kidney disease related to lupus and general lupus, which were not successful. And one of the thoughts as to why they were not successful is that the drug rituximab didn't achieve enough B cell depletion. So can we develop treatments that allow for deeper or depletion of B cells? Can we do a better job of getting rid of the B cells, if you will, without harming the patient? Next slide, please. So belimumab depletes B cells to some degree, but not to the degree that rituximab does. So here again is the B cell, okay? And then I talked about the receptor called the CD20 receptor, and rituximab attacks that CD20 receptor. The B cells grow in part or get stimulated by a factor called B cell activating factor. And what belimumab does, so belimumab works differently from the way in which rituximab does. So rituximab attacks the B cell through this molecule and directly depletes the B cell, whereas belimumab or Benlysta works indirectly to deplete the B cell. It attacks this growth factor, this this molecule that allows for the B cells to grow. So belimumab is an indirect depleter of B cells, and rituximab is a more direct depleter of B cells. So rituximab does this very well. There's another drug that's been developed that looks like it's even a deeper depleter of B cells called obinutuzumab. And then there's cellular-based therapies, which are great depleters of B cells. Next slide, please. I'm not going to go over this, but this is just lists all the biological therapies in lupus that affect different components of the immune system. Okay, next slide. I'll leave that for you. And so I just want to leave you with the concept of deeper B cell depletion. I've shown you how B cells are involved in the pathogenesis, how lupus develops in the human body, and how B cells are important, and B cells interact with other cells of the immune system to produce autoantibodies that then result in inflammation. And so getting rid of the B cells or allowing for more or deeper B cell depletion is a potentially more effective way of treating lupus. And CAR T therapies, which is going to be discussed with the next speaker, will involve concepts that that illustrate the importance of deeper B cell depletion. Thank you for your attention.
Excellent. Thank you so much, Dr. Maserati, for really showcasing how B cells are important in the pathogenesis of lupus, and certainly how work has been done to target B cells. And because lupus is such a complex disease, and there's so many different inflammatory pathways involved in lupus, oftentimes efforts have failed. So now Dr. Caricchio is going to give us an overview of CAR cell therapy and tell us about the potential of this therapy to really be effective at targeting B cells and certainly improving outcomes in lupus. So we're fortunate to have Dr. Caricchio, who's the chief of the Division of Rheumatology, director of the Lupus Center at the University of Massachusetts Chan Medical School. Welcome, Dr. Caricchio, and I hope I'm pronouncing your last name right. I know you're probably over it with us messing up your name. So if you want to, for the purposes of
No,
correctness,
no,
tell us.
thank you,
That's perfectly fine with me, but welcome. It's such a
Thank
pleasure to have you on our platform this evening, and looking forward to your talk.
Thank you, Joy. Now that was close enough. That was close enough. It's understandable, and but of course it is. It's quite a pleasure to be here, and I see almost 300 participants. So obviously there is a great interest in the effort that the LRA is doing. So if you don't mind proceeding, and also I'd like to thank my colleagues for their talks, who, thanks to theirs, they made an easier job for me to present my presentation. And a couple of disclosures about who I am. I do consult for a number of companies to try to help them design clinical trials, but also involved in a large network of lupus sites, lupus clinical sites that also participate in a number of clinical trials and are directed by, in this case, the Lupus Research Alliance. And the group is LUCIN, so-called, LUCIN, Lupus Clinical Investigator Network. Having said that, if you can proceed, please. Yes. So I do like to show this slide. It's an exercise that used to be done. Now it's no longer possible, but it used to be done in a website that the audience, if they're not aware, should write down. It is called clinicaltrials.gov, and it's listed down there. This is a place where run by the federal government, by the NIH as well, in which all the clinical trials undergoing on the planet are listed, and one can search every single one of them with the inclusion and exclusion criteria by continent, by region, by state, by even city. So one can really look into all the clinical trials. And up to a few months ago, it was possible actually to generate a map of the world to see where those clinical trials were going. And so the exercise that I've done is to just search for all the clinical trials in lupus, plus few other systemic autoimmune diseases, such as scleroderma, dermatomyositis, or inflammatory myositis, and multiple sclerosis. And if you can click, please. And what has emerged that in a span of 2 years, that is 2 years search, 2 years ago, there were only 10 trials worldwide, a year ago, 14. Now, a few months ago, 6 months ago, roughly, there were 42. And then a month ago, I did the same search. And if you advance, please. And in a matter of a few months, the trials have doubled. Now, those are all the trials in the world that are specifically linked to CAR-T, clinical intervention in lupus and similar connective tissue diseases. So the trials are hundreds of thousands, but the specific ones on CAR-T are 96. And not only that, but you can see how exponential the growth of those clinical trials has been only in a span of 2.5 years. Now, admittedly, the CAR-T therapy is not an invention of rheumatologists or lupologists or nephrologists. It does come from the world of hematology clinical research. That's where it started, and that's where we are taking it from. But that's where the innovation in cellular therapies have come so far. And so if you do the same exercise in the hematological world, such as if you search for CAR-T in leukemias and lymphomas and some few solid cancers, which are by far a minority, if you advance, you'll see that the number of trials in the last few years is now getting into the 1,000. So if you can imagine that right now they're driving the innovation in cellular therapies. And so if you think that there are 10 times more, then you can maybe envision that a few years from now, we will have quite a number of options for our own lupus patients. Please, next. Next slide. Thank you. Now, that didn't take 5 seconds. It took actually quite a long time. This is a bit of a history of this type of intervention. Now you can see that it took almost 20 years. Those terms that you're looking at, double chain of TCR or single chain or lymphodepletion in 2002 or incorporation of what is called 4-1BB, but it's a co-stimulatory molecule. So it took 20 years to actually figure out and understand how to build this really marvel of molecular biology engineering. So it took almost 20 years just to do that. That was 2004. One click. And it was in 2006 that the first patient was treated. And not only that, but it was just a few years later that the first patient with leukemia had a complete remission. And in 2012, rapid succession now, was the first patient treated with pediatric patient with leukemia, which is still in remission. And in 2007, only in 2017 was the first one approved for leukemia by the FDA. Kymriah is called. So you see the span, it's quite remarkable. So that's, if you do the math, it's 30 years. So it took 30 years to get to the first approved medication. And it was only, please, next click. And it was only in 2021 that the first patient with CAR-T was treated with CAR-T that had lupus. So it feels, at least for those heavily involved in the CAR-T world, and also that have treated patients or they hear about CAR-T, that has been a decade that is around. But actually, the reality, at least in lupus, it's only a few years that the first patient was treated, which is great. Now we have so many trials ongoing. Can you please go ahead? So never underestimate how long it took for these very, very important investigators to get to the point to create something that has been safe so far. Now, what it is? What is this chimeric antigen receptor that we've been using in leukemia for the hematologist and lymphoma, and now in connective tissue diseases, and in particularly lupus? Can you click for me? So the chimeric antigen receptor, what it really is, it doesn't exist in nature. It is a molecular biology creation that is then made expressed by cells of the patient, him or herself, or from a donor. But what it does, it takes advantage of the two best ways we have to defend ourselves from infections and also from cancer, at least initially. So it takes advantage of what you see on the left, the way that T cells, a major arm of our immune system, that have to recognize if there is an infection around, or if there is a cancer. And the one on the right instead is our monoarm antibodies. So the way that we have actually to recognize also infections and tell the rest of the immune system to attack that bacteria or viruses and eliminate. The chimeric antigen receptor, can you click please for me, is actually a fusion of those two natural ways of defending ourselves from infections, and that makes it what's called the CAR-T. So they took part of the T cell on the left that is able to activate when there is an infection, and they took part of the antibody that is able to recognize the type of infection and fuse it together. Now, the beauty of that is that you can make that chimeric antigen receptor targeting anything that you can think of. And for the most part, those chimeric antigen receptors have been made to destroy B cells, the cells that in leukemia lymphoma generate the leukemia and the lymphoma. But in autoimmunity, conveniently, are the cells that generate the autoantibody, as supposedly the lupus and other connective tissue diseases. Can you click, please, for me? Did you click? Yes. Now, no, only once. I want to give credit. Can you go back? Because there was something else. Now, on your right, what you see, you don't have to learn anything, but what I wanted to convey is that this protein, which is engineered to be expressed in the cells of the patients with lupus or from a donor. I'll go briefly through that. It is actually very complex, and every single piece has gone really excruciating investigation to make it really perfect to do its job. So it looks like very simple at some point, but actually it took an army of scientists and translational investigators to actually fine tuning every single part of these mixed of proteins. Now you can go, please. Now, why then there has been an explosion of interest in lupus and other autoimmune diseases? Now, what you see on the left is a cartoon of what cancer might be. Cancer and the white cells, the one gray light, are the cancer cells. They are a lot when it comes to cancer. And there are only few cells that are able to control them. But it's also, the cancer environment is also a very inflammatory environment, very difficult also to penetrate. On the right, on the other hand, you're looking at a normal lymph node with few more cells that are responsible for lupus in this particular case. And it's not really a very strong inflammatory and difficult to penetrate tissue. So the CAR-T, in theory, it has a better chance of get to the target and destroy it and kill those B cells. And for that reason, can you click for me? There has been an explosion of interest in many autoimmune diseases, and we are here to talk about lupus. But lupus is by no means the only disease that is being targeted with this modality. Can you go ahead, please? More. I have a lot of clicking. Now one aspect of this type of therapy is that it comes in a variety of ways to be delivered, and those are three very complex, each one of them, three modalities that take advantage of the advancement of the molecular biology currently and are able, each one of them, to deliver into the cells of the patient that is going to be treated this chimeric protein, this protein generated, engineered in the lab, so that those own cells of the individual, or in other cases from donors, can be reinstated in the patient. They proliferate and they search and destroy, so to speak, the target, which is the B cells in this particular case that are supposedly generating the disease, the lupus disease. There are different modalities. I cannot point at them because I don't have the power on the pointer. But those are the three major modalities. Can you click again? Now, there are a major point is that it can be directly generated in the cells from the patient and then given back to the patient, or, and they're called autologous, or they can be created from a healthy donor in the same fashion and then given to the patient without being destroyed, but by doing their job, and then in that case are called allogeneic. And then finally, there is a third modality that is directly in vivo by creating a molecule that in giving it to the patient, the cells of the patient inside the patient actually express the same chimeric antigen receptor. And by the way, I didn't mention anything about the term chimeric for those perhaps that do not know, but this is an ancient Greek word that comes from the ancient Greek mythology, in which the chimeric, the chimera, it was an imaginary animal, although not so good looking, an imaginary animal made by different types, made by parts of different type of animals. That's why chimeric, because it comes from B cells and T cells items. Can you go ahead? Now, that is not the only thing that is complex about these chimeric antigen receptors. The other complex things is that they don't have to come from a single source of cells. There are a number of different types of cells that can be engineered and then given to the patient to be so powerful in destroying, so to speak, lupus cells. Can you go ahead? And that is not just this is now, but there are also coming new methodologies to make them more precise, more durable or less durable, depending on the situation. So what I wanted to convey is that this is, even though it took 20 years to make it, the molecular biology of it is growing so fast that there are more and more different versions of the same iterations of the same methodology. Can you go ahead? Having said that, we do need to prove the ones that we have currently that they work. This is to show you just the yellow part on the left, that these chimeric proteins, for the most part in lupus trials and similar conditions, are targeting two major molecules, which is the CD19 and the BCMA, which my colleagues, especially Dr. Massarotti, showed that are present on B cells. Those are markers of B cells, and that's, for the most part, those therapies have been utilized in lupus. Can you go ahead, please? One more. Now, speaking for the autologous one, which is the product, the type of CAR T in which the cells are taken from the patient, then in vitro transfected with this new protein and then given back to the patient. The protocol in this particular case, the treatment protocol, is complex. Can you go ahead? And it has several phases. The first phase is the fact to collect the cells from the patient, a significant number, and it's called apheresis or leukapheresis because it specifically filters the white blood cells, the lymphocytes in particular. And once taken from the patient, there is the next step, the next 3 steps is quality control, a lot of quality controls actually. And then the engineering, so the transfection of those cells that then are going to be able to express this chimeric antigen that will target the B cells in lupus. They have to be expanded first in vitro, and then eventually the last step is to reinfuse them in the patient. Now, it has been shown a few years ago, 20 years ago, that lymphodepletion is something that helps in this particular protocol for the transfected cells given to the patient to expand. And so the patient, before undergoing the restitution of her own cells to expand, the patient undergoes certain treatment, which is called, which is composed of fludarabine and Cytoxan. It's a combination that is very typical for those treatments, although there are higher level in cancer, leukemia, lymphoma, and lower levels in lupus and other connective tissue diseases. And at that point, all the cells expand, the cells that have been transfected expand, and search and destroy in the tissue the B cells that are responsible for lupus. Can you go ahead, please? And the CD19 is one of the major target that is being developed for those CAR T. And what happens once the cells are given back to the patient, those CAR T expand, multiply many times. And what happens is that they search all the CD19 positive marker positive cells in the tissues, in the kidney, in the skin, in the liver, in the lung, wherever they are, and they just kill them. And this is to illustrate that when they kill them, they kill a wide variety of the different B cells. We have a lot of different B cells, and those type of interventions kill them in a significant wide variety. Can you go ahead? And this is just a cartoon that the thought about the CAR T versus an antibody, such as Rituxan, for example. In the bottom, you see a Rituxan treatment, which still leaves some B cells alive, while the CAR T is supposed to, in the tissue, to deplete all of them, and then wait from the bone marrow to then reconstitute. Can you go ahead? And this is it's called an histology, but it is a demonstration of how deeply those CAR T kill those B cells that are responsible for lupus. What you're looking here is an histology, and you should concentrate on the pictures that looks like brown, because brown means presence of B cells, lupus B cells in the tissue. And then the first row is from a patient that before receiving CAR-T. And there is a lot of brown indicating a lot of B cells infiltrate. After CAR-T, 3 months later, this is from lymph nodes, there was no B cell to be found. And post instead, the last row is instead after receiving Rituxan, showing that in a lymph node, even though in the periphery, Rituxan deplete B cells in the blood, but when you look into lymph nodes, there is still a number of B cells, indicating as for why this intervention that targets the same molecules is much more powerful. Can you go ahead, please? Now, it doesn't come for free because every good treatment, every excellent treatment does have the potential of side effects. The first most common, although the first most recognized, I would say they're not common, are the ones that you see on the left, but we will hear more about them, so I don't want to spend too much time, but it's the cytokine release syndrome, the immune cell-associated hematotoxicity, and immune effector cell-associated neurotoxicity. Depending on the type of CAR-T, some can occur, others cannot at all. But so far, those have occurred, but have been all quite manageable because we have learned from the hematological world how to do that, and they're pretty good at managing those adverse events. But again, we'll hear more from my colleagues. Can you go ahead, please? Now, Dr. Massarotti very well, unfortunately, expressed our numerous failures when it comes to depleting B cells, and then here are pretty much the same iteration of the B cells and the approaches that have been tried so far with relative success. Can you click one? And those are the failure the fact that those interventions have not worked when tested in lupus, despite they were all targeting the same marker, which is CD19 or CD20 or similar anyway on B cells. Can you go ahead, please? And just to recognize who thought early on what to do with CAR-T, this is actually a mouse work from Marko Radic, who still is a mouse investigator. Actually, he had the idea of thinking, what if we use this CAR-T that was so successful in leukemia and lymphoma that targets B cells, which are very important in lupus? What if we try them into a mouse model that resembles lupus? And what is illustrated here, when you see the bars going down in this particular case, is the survival of these two different types of mouse models. But when you look at instead at the bar that keeps going over in terms of survival, that's after these mice have received the mouse equivalent of CAR-T. So it was quite a successful demonstration that this intervention might have worked, and has in part inspired Georg Schett to also, which was the pioneer in Erlangen, Germany, to treat the first lupus patient, to go ahead and do something like that. Can you go ahead, please, for me? And that's him on the left, on your far left. I was visiting there with my colleague, hematologist. And what you're seeing, there is a young woman there who's a molecular biologist now, at least studying for that. She is the second patient that he treated a few years ago. And when I went there 2 years ago, she was already 2 years out of treatment, and she was doing quite well. And it was quite emotional because nobody has ever seen anything like that when it comes to treatment with lupus. And he was, of course, off all therapies and eager to continue her studies. Can you go next, please? One more. And then we also well, at least in the scientific world, we also this data from the first few patients that were treated. Now, for the patients or relatives and caregiver in the audience, you should focus on the first 3 lines where you see 1, 2, 3, 4, 5, 6, 7, and 8, and SLE, and the term DORIS remission. Now, DORIS remission is a definition of remission in systemic lupus erythematosus, which is what it means. But it means that the patient has to be off treatment, doesn't have any sign or systemic or symptom of lupus for a certain period of time. And all the 8 patients from the first group in Erlangen reached that that status, which was unprecedented and is still unprecedented. No treatment is able to do that yet. And that was 2 years out of treatment. The other thing noticeable that it wasn't that so achievable before, or at least even for now, is the fact that any measure of lupus disease activity in those 8 patients went down, was no longer there after 3 months from receiving the CAR-T cells, all autologous in Erlangen. There was from disease activity, which is the SLEDA, it was for the anti-double-stranded gone, and also for the what we look at as inflammation when it comes to complement. Complement usually is low in a number of lupus patients, but in these patients were all normalized, and including the proteinuria, so the protein in the urine, because all these patients had lupus nephritis and their proteinuria just resolved. Can you go ahead? We won't stay here, but this is for other 2 types of autoimmune diseases, scleroderma and myositis. Similar results, early results, but similar. Can you go ahead, please, for me? And it wasn't just as a demonstration, a change in the disease manifestations, meaning that they couldn't find the disease, including remarkably the fatigue. Those patients were all fatigued, and fatigue went away as well. But it is also in looking at what is coming back after the treatment of these patients with the lymphodepletion and the CAR-T. What comes back for now in this patient looks like a new immune system that is no longer autoimmune, like a fresh restart. And that's because that is the way why, and that's the reason why there is this concept of a reset, lupus reset, because it looks like that after that significant drop of B cells that were killed by CAR-T, including partly for the lymphodepletion, which will also drop the T cells. Then what comes back several weeks within 2 months later is a new fresh start, so to speak, for this immune system. And that is also the remarkable part, is also the fact that the interferon signature, which is something that we all think is very profoundly important in lupus disease, also has a part of a reset. Because there is much less interferon signature in these patients. Can you go ahead for me, please? Now, the point here is that the CD19 is the major target, but there have been also interventions with the BCMA. The BCMA is a marker of the cells that actually produce the autoantibodies, while the CD19 is more a marker of everything else up to the point that those cells become able to produce. And the combination seems to be also effective. This was presented last year at the American College of Rheumatology, in which a number of lupus patients were treated, 13 in particular, and all had at least 6 months had resolution of their lupus manifestation. Interestingly, 2 of those actually had lymphoma, and they were treated also for lymphoma. And of course, not only lymphoma was treated, but also lupus disease. Can you go ahead for me? And this is all here. When you see all these values going down, means that those patients were doing fine. And then the important part is that those are all autoantibodies, which are produced by the bad B cells in lupus. And they all went down very synchronously. Can you go ahead? How many slides there? So what about in our current trials, and if we could compare them? So this is what you see. They look like that the anti-DNA went down from this clinical trial that I just showed you, and the complement went up. Very typical for those clinical trials and in lupus when lupus is quiescent. Next. And another clinical trial, which is this one from Caverna, it looks like also similar. Everything goes down in terms of autoantibodies, but the complement also become normal because it's a sign of no longer disease. Can you keep going? And this is the proteins in the urine from the patients from Erlangen, Germany, in which also they go down. So there is no longer proteinuria in these patients because there was a resolution of the renal disease. One more. This is not exactly the case from the other patients. Some patients still had proteinuria or after treatment, or the proteinuria went up and then down again. And the thought is that it's not because it didn't work, but because most likely the patients in other clinical trials had already some damage in the kidney. And so damage in the kidney, unfortunately, is yet to be able to be reversed. So the reflection of these proteins in the urine is more of a damage that will have some proteins in the urine rather than lack of efficacy in the kidney. Go ahead. But all of them were nevertheless drug-free, which is quite remarkable, except for one recurrence in Caverna trial. Can you go ahead, please, for me? Now, I mentioned that this is not the only one. There is the autologous, which is your own cells or the cells of the patients engineered in a way that can kill those B cells and give it back. But there is also the allogeneic, in which there is a donor, actually. And one healthy donor, the cells are taken, and they are then engineered as the killer cells of lupus-specific B cells in the individual with lupus. And he offers several advantages, which we'll hear shortly. Can you go ahead for me? Yeah, so there are at least two types. The iPSC, which stands for inducible pluripotent stem cells, or the second one is NK, natural killer CAR-T. They both do the same job, except that they come from a donor, a healthy donor, and they can be expanded into hundreds of doses, actually, rather than the simple one, the autologous one, and they're also ready to go, meaning that they are off the shelf, so-called. Once they are generated, they can be frozen and then given to the patient that is in need. And they have also this term of universal CAR-T because they can be given to anyone. Why? Because also there is another molecular engineering feat. Can you go ahead? And the reason is because those cells, they can come from two different sources. Either, and they are from a healthy individual on the first group of images on the top, they can come from two different sources and made with a soup of stimuli into iPSC, into inducible pluripotent stem cells. Those will be transfected with the CAR-T, the CD19 killer, so to speak. But once they're made with the CD19 killer, they have to go through also the masking, so to speak, of the fact that they do not belong to the patient, but they belong to to a donor. And our immune system is very smart. But I guess molecular biologists are smarter. And what they did, they delete everything that could make those cells be recognized by the immune system of the lupus patient, and then they get injected. And then they get injected, and then they search and destroy the B cells that induce lupus. The advantage is that they're ready to go, meaning that they're off the shelves. They're waiting for the patient to be received. There is not really time for creating them. They can be redosed, and they and some of those clinical trials are not using lymphodepletion before treatment to make space in the individual who receives them, and then potentially there are fewer side effects. Can you go ahead? And this is one example in which they did not treat lupus, but rather dermatomyositis and scleroderma. But in both cases, well, both dermatomyositis actually, but in both cases they were successful. And dermatomyositis or systemic sclerosis are relatively similar to lupus, being all of them connective tissue diseases. But what they did, they took cells from healthy donors, they transfected with the CAR-T, then they delete everything that could be recognized by the host immune system and then infuse into the patient. Can you go ahead? And those two patients were actually received a good response. This is another example. Again, 20 patients presented last year at the same American College of Rheumatology. In this case, though, they did use donors, but they did use the lymphodepletion, and for now, up to few months, 6 months precisely, a significant number of patients have received the cells, and they are in remission, 50%, while 75% are actually in what we call low disease activity state, which is quite strong. Nevertheless, result. Go ahead, please. Okay, and this is, I think, the last one, and it's interesting because CD19.
So it looks like we may have lost Dr. Carricchio. He is frozen, but I believe this was likely his last slide. So thank you, Dr. Carricchio. Are you here?
Can you go ahead one more?
Okay, back.
And so, no, one more. And so that's what you're going to hear from, which I'm very privileged to have treated her. And then you can go ahead, one more. And I thank everyone.
Thank you so much, Dr. Carricchio, for that phenomenal presentation and breakdown of the different types of CAR cell therapy that exists, and the results that we have thus far with these treatments. And you can see all of the celebration and excitement coming through. Next slide, please. So next up, we have Dr. Ahmet Saxena, who's going to be continuing this discussion. Dr. Saxena is Associate Professor of Medicine, Department of Medicine in the Department of Medicine and Division of Rheumatology at NYU Langone Health. He's going to be talking about T-cell engagers as well as other types of CARs and how they're a little bit different in nature. Thank you, Dr. Saxena, for joining us this evening.
Sure, thanks for having me, and thanks for everybody for sticking out through this. And I'm hoping everybody's learning a lot. And I'm going to talk about some potential next steps, which is kind of crazy because we're really still in the infancy of studying, CAR therapy in general. But already people are starting to think about what can we do as the future, how can we make this even better? And so there's a couple of things that are in the pipeline that I'm going to talk to you about the science and what little data we have so far, but at least kind of whet your appetite for the future. So you can go ahead to the next slide. So Dr. Carricchio mentioned this a little bit, but I'm going to dive in a little bit more into something that's called the natural killer cells and how we could use those cells as a different type of predator for destroying those B cells, which are, our target that we're trying to get rid of in lupus. And so natural killer cells, they're a different type of lymphocyte than the T cells that we talked about. They're actually part of something that's called the innate immune system, which destroys both infected cells and tumor cells without the need for other cells to activate them. Those T cells require activation to get going and destroy those cells, but these natural killer cells don't. And they also don't really need any prior exposure to the target. It's not like you have to have, seen a bacteria before to remember it and then be able to fight it off in the future. These natural killer cells can do that damage, without having ever been exposed to a target before. Because of that, they're less specific than T cells. They kind of affect a wide variety of threats, other cells that are just kind of blanketly infected or kind of, general tumors, for example. But they kill cells quickly. They do it often in minutes, which is really where they get this fantastic term, the natural killer cell. And over on the right here, you can see it in all of its spiky glory, looking like it's ready to get rid of some of the bad guys. And what happens though is, in addition to just killing the cells themselves, they also release some signals which activate some of those other immune cells to help get rid of the bad guys. And won't go into too much detail, but you can see here on the bottom what we're just looking at is the way that these cells actually work. And on the left, if you've got your own healthy cell, it expresses some signals that say, hey, I'm okay, I'm me, we're us, don't come and get me. And so that's on the bottom here, this MHC class 1 receptor, which tells that natural killer cell, not to destroy the healthy cell. But if a cell has been infected by a virus or maybe is a tumor cell, sometimes you'll lose that self receptor, and the natural killer cells will then know to destroy that cell. And other infections might actually induce a lot of activation of the natural killer cells, telling them, hey, this is a bad guy, this is a foreign cell, let me come and get me, essentially. And so that's the way that natural killer cells work. You can go on to the next slide. And the differences with how natural killer cells work versus how T cells work allow for some potential differences when creating these CAR constructs, when you're coming up with how to actually use cells to destroy B cells. And so the CAR NK cells, they don't require compatibility with the host. You don't have to be getting these cells from yourself. And so for that reason, these fall typically under that allogeneic basket that Dr. Krieger was mentioning. These are those off-the-shelf products that you can use without having to stick a catheter in somebody's neck and take a bunch of blood out before it gets genetically modified and put back in. And these are engineered from donated blood products. They're really never coming from the patient themselves. And they're also NK cells are not really activated by mismatches. These cells don't say, oh, I don't like that you're, foreign from a different person. So these cells don't end up attacking the healthy cells in your body, which is something that's called graft versus host disease. That's an issue that we haven't really seen too much with CAR T in lupus, but has been something that's been noted in patients when they're treated with CAR T for cancers. And so the theoretical lack of that issue is only a positive with these types of constructs. They do still require the traditional chemotherapy before this therapy. And that is used to essentially make that space in the immune system and allow the CAR cells to activate. So you do still need that. That is similar to many of the CAR T constructs. But when these cells are in your body, they expand less than the CAR T. They don't multiply as much. And so that could lead to less of the side effects that we see with CAR T, some of those overactive inflammation CAR T side effects, which are called the cytokine release syndrome, or this ICANS, which is something that can affect the central nervous system. So there could be less of those side effects with this therapy. Next slide. Also, these cells don't really hang out in the body for as long as the CAR Ts do. And whether that's good or bad, we actually don't really know. We don't know if you need to have these hanging around to keep the immune system in check, or if we can just do this once, get this expansion quickly, and then hopefully reset the immune system. These cells are cheaper to produce. They're faster to use than the CAR Ts. Although there are still some obstacles in how we store these products, and I'm sure things like this will get worked out as the science moves forward. The other thing about CAR NK versus CAR T is that actually there are no real CAR NK treatments that are actually approved yet for cancers. They are being studied in cancers, but nothing like the CAR Ts where there are a bunch of already approved therapies. The studies in lupus that are using the CAR NKs are still targeting the same CD19 B cell lymphocyte targets as we've seen before. So that's another similarity. Next step, next slide. So this is actually the same data that Roberto just mentioned, but this is that first reported CAR NK data out of China. These 20 patients with active, severe, refractory lupus. They received 3 doses of this CAR NK therapy after getting that lymphodepletion with those chemotherapy agents, cyclophosphamide and fludarabine. They studied 3 dose levels, and the median follow-up was 5 months. And there was only a very minimal side effect profile. Only 2 patients had short-lived fevers. And as mentioned before, of those 8 patients that had been followed for more than 6 months, 50% achieved remission, 75% achieved low disease activity. And the biggest point was that complete deletion of B cells and the reset of the immune system was achieved. That's the hope, that reset of the immune system could get us towards that, vaunted C word of the cure that we're all hoping this therapy might do. The B cells did reconstitute within 2 to 3 months after treatment. And so these cells do come back. They just come back in this kind of fresh, new, naive fashion that is hopefully telling us that the immune system has been reset. Next slide. So there are a couple of currently enrolling CAR-NK trials in the USA, and we're not going to go too much into detail, but they each have their own kind of factors that make them a little different from each other. But they're both looking at lupus patients. This one, NKX019, is looking at patients with refractory lupus nephritis, receiving 3 doses after lymphodepletion. And this other one called AB101 is looking at this modified NK therapy in combination with a couple of the more traditional B-cell depleting therapies that you may have already heard about and that Dr. Maserati mentioned, like rituximab. And so, these trials are enrolling and they're available, and we'll see, how they do along with the other studies that are being done with the CAR-Ts. Next slide. We'll move on quickly to the bispecific T-cell engagers, or what we call BiTE therapy. And the question is this actually the future of cell therapy? Could this give us the benefit of everything we've heard about so far without some of the limitations? So BiTEs are lab-engineered monoclonal antibodies. And you've probably heard of that term before because a lot of the therapies that you know in lupus, like belimumab and anifrolumab, are also monoclonal antibodies. And similarly, these are given either by subcutaneous or intravenous infusions. And the way these therapies work is they've kind of got 2 arms. And one of the arms will grab on to your own body's T cells, unmodified, not genetically changed T cells, just the ones floating around in your blood. And then the other arm will grab the B cells that they're trying to destroy. And essentially, they'll bring them together. They'll bring them next to each other and allow for the T cell to become activated and release the same type of proteins that are used in the CAR therapy to destroy the B cells themselves. And so it's using your own T cells to destroy the B cells. And this has already been approved in cancers. There's several agents approved for blood cancers. And they tend to result in the same type of immune reset that we've seen in the traditional cell therapies when we're targeting the B cells. Next slide. So there have only been a couple of case studies using these types of treatments in autoimmune disease patients. One of them, which I'm not going to even attempt to pronounce, was used in rheumatoid arthritis. And it looked at 6 severe rheumatoid arthritis patients that had been refractory to multiple medications. This one was given as an IV infusion, and it had very good clinical responses without significant side effects, and again showed that immune reset, which we were looking for. This other one, which targeted that other B cell marker called BCMA, as well as the T cells, because remember, these are bispecific, they target both the B cells and the T cells, is a treatment that's used in myeloma already, but was studied in one patient with lupus nephritis who had failed multiple medicines. This one was given subcutaneously 4 times in 5 weeks. Other medications were stopped, similar to what we see in the CAR-T therapy. The patient had a little bit more of the inflammation side effect, this CRS, some infections and low immunoglobulins, showing that the immune system was reduced. But there was a good clinical response starting 4 weeks after treatment. And there were also good responses in patients who had scleroderma, Sjogren's syndrome, myositis, and rheumatoid arthritis as well. Next slide. So what are the pros and the cons of these therapies versus what we've already talked about with CAR-T? So the advantages are that you don't need to take the blood out and genetically modify it. That's the apheresis. We don't need any of that. This also is off the shelf. Probably most importantly, no chemotherapy, no lymphodepletion required. There are some constructs that are going to be trying to do that as well, but this one by definition would not require that chemotherapy, which is really one of the most limiting things when we talk about why we might not bring this therapy to everybody, because we want to avoid that potential risk. So if we can avoid that risk, that'd be pretty impressive. Potentially less frequent side effects of that inflammatory diseases, and the potential to adjust the doses if you need it. Anytime that there is some genetic changes that are done, there's always a question of whether you might end up with secondary cancers, but that's something that you might not see with this type of therapy. And you'll be able to easily produce this and potentially redose it if you have a relapse. But on the flip side, in cancers, these therapies have not been as effective as CAR T. Now, do you need to be as effective in lupus as you do in cancer? Maybe, maybe not. We're going to find out when we do these studies. And they may require repeated treatments. And one of the big things about CAR T that everybody loves is the idea that maybe you can get one time treated and then never again with your life. And so that's one thing that might be a disadvantage if you needed to get this over and over again. Next slide. Just to summarize, while we're still studying the safety and efficacy of CAR T in lupus, there are other mechanisms that might work similarly with less intense therapy and side effects. The CAR NKs we talked about might offer less off-target side effects. They're easy to produce. The BiTE therapy can be given without chemotherapy and could potentially be redosed with flares, but they might need ongoing treatment. And obviously, further studies are required to evaluate the efficacy and safety, which is what everybody's working on right now.
Excellent. Thank you so much, Dr. Saxena, for that wonderful presentation. And now we're going to skip ahead to Ms. Talea Reed, who is our featured patient for this evening. Talea has been living with lupus for a while now and had the courage and the support to enter into a CAR T-cell therapy trial. Her treating physician, Dr. Caricchio, is also on the call. So I want to turn it over to Jaron Crump, who is our grants manager here at the LFA, and he'll be asking Talea a couple of questions. Talea, thank you so much for joining us this evening.
Hi, everyone. Hi, Talea. Thanks for joining us again. It's so great to see you.
It's good to see you too. Thank you.
Yeah, so I want to just kind of jump to some of, I'd say, like the high points of the conversation, the interview, that we're having here. Could you share some of what your experience was like living with lupus prior to CAR T-cell therapy, and then maybe explain some about how things are different for you overall since you completed it?
Of course. So before I received the CAR T therapy, I was experiencing like a lot of joint swelling, a lot of pain trying to move and do normal things throughout the day. Also, things like rashes were appearing on my face, my arms, areas like that. And I was very fatigued. So it was hard for me to get up and go to work or go to school. So it kind of made me not have, like, I guess, any normacy. After receiving the treatment, I would say like it was a complete 180. When I would go to sleep before, I would wake up and still feel tired no matter how long I slept for. So then after I received the treatment, the fatigue actually went away. So I felt like I had more of a day compared to before. And also after the treatment, I didn't have to have any lupus medications. So that definitely was a nice change to experience after having lupus for so long.
Thank you so much. I know fatigue is really, one of the main things that people talk about when they're dealing with lupus. Could you walk us through a bit of what the process was like when you were receiving therapy and maybe some of the biggest challenges that you might have experienced while you were going through it?
Hmm. So when I started the process of getting to receiving the therapy, I had to have my cells taken out, and then they sent those out to be modified. Um, then we had to wait for them to grow to the amount that they needed to be at. Once it became the correct amount, they were sent back. And a week prior to receiving the cells, I had to get off of my lupus medication. Um, and then I also had to go through chemotherapy to try to get the cells count in my body lower so that it could make room for the new cells that I was receiving. That was for a few days. So then after I received that, the chemo, I was able to get the cells reinfused into me. And then from there, I just kind of just went through, like, I guess my body kind of reacting to the new cells that entered, and the cells were working to try to get the lupus to calm down and go away.
Thank you so much for that. Um, were there like any specific moments throughout the process that really stand out to you that you think you'd like to share?
Um, I would say I liked how every different area, I guess, of the hospital that I was at, the different areas of like expertise, they all communicated with each other. Um, so it was like rheumatology, oncology, infectious disease, etc. They all kind of monitored me to make sure that after I received the therapy, that everything was functioning correctly and that I was okay.
Okay, and if you were able to give someone who's considering CAR T cell therapy some advice, what would you tell them? And also, what's something that you wish someone might have told you before you did it? And is there anything like unexpected that happened once you were able to, finish?
So advice that I would give someone who is looking into getting the CAR T therapy, I would say ask questions. It's good to know exactly like what's all entailed, what's expected, what could happen. That definitely can help the process so then you don't feel like there's any surprises. Um, I guess maybe something that someone could have said to me was relax. I think that I didn't know what to expect, so everything was unexpected to me. Um, so I think that if anything, I guess was off, or maybe I didn't feel well, I automatically thought like, oh my gosh, something's bad is happening, or but I think I just needed to relax and that it was gonna work out in the end.
And what would you say like led you to your decision to participate and to, choose this therapy as an option for yourself?
Um, so I would say the fact that the medications that I was taking prior, um, they weren't really working for me. I still was experiencing like pretty bad flares, still hard to like get up and do what I needed to do throughout the day. So I kind of felt like I was at a dead end and I really wanted to try something new that could possibly work and like help calm down my lupus and get me off of like the medications, at least half of them. I'm grateful that I've gotten off of all of them. So
How would you say you manage the emotional and the, potential physical challenges when you're going through the CAR T therapy process?
it definitely was a change. Without my family, I think that it would have been harder. They definitely have been there from the beginning with my diagnosis. If I didn't feel well or if I couldn't do something, they jumped in and they helped me out. And that was no different when I received the therapy. They were my biggest spokesperson if I didn't feel well, or they were there just as much as the doctors and the nurses. They were there too.
Yeah, and I know you already mentioned just the fatigue and how that changed for you after you finished the therapy. Would you say that that was the biggest impact of CAR T therapy for you, or is there anything else that really stands out as like just a big significant change to you and your overall quality of life since you did it?
I think the fatigue and also like the joint pain, they both were the main things that I noticed. Because it just affected me so much. Even simple things like driving my car, which doesn't, you know, have that much function or anything that you would need, but I still could tell like, oh, this is uncomfortable, or I don't know if I could do this today, and kind of had to hype myself up to do certain things that would seem normal to other people.
Thank you so much, Stella. It's really great to have you here and to have Dr. Caricchio here. I'm going to turn it back over to you, Dr. Bowie. I know that there's a few things that you wanted to share before we reached 8 o'clock. Thank you again, Talea.
Thank you.
Thank you so much, Talea, and thank you, Jaren, as well. There is one question for you, Talea, in the Q&A, and it's, what lifestyle changes did you experience after getting CAR T-cell therapy? Did you religiously have to put on sunscreen and cover up?
I would say that most people should put on sunscreen. I don't think that I would say that I have to put on any more or any less than anybody else. I guess as far as changes, since I have more energy now, I feel like I exercise a lot more than I was able to before. Especially like when I would get sore before, I feel like the soreness would last way longer than it normally would if I were to exercise now. So that's definitely a healthier lifestyle change that I experienced.
Awesome. Well, thank you again for sharing your story. And if we had time, I would have Dr. Caricchio chime in from the physician's perspective. But we have a few items to share before we close out this evening. So thank you all again.
Her. I'm texting Talaya, so I'm being charming personally. Yes. They did a wonderful job, wonderful job.
Perfect. Safo, if you could go ahead and share our closeout slide for us, that would be great. So again, thank you to all of our speakers, to all of our sponsors, and to each of you who have joined us this evening. We do have a few things that we ask of you before we close out this evening. You'll see on the left side of your screen a QR code. If you could go ahead and take out your phone, open that camera app, and go ahead and scan that QR code for us. That's going to take you to a survey. Filling out that survey is imperative to making sure that we can continue to bring this type of programming to the community. We're always looking at ways that we can improve what we offer and how we offer it to the community that we serve, and that's you guys, because we want to make sure that we're doing a good job. And the only way we know that is through your feedback. So please take some time. It's a really short survey. Please take some time to give us some feedback and let us know what additional information you'd like, and we'd be more than happy to oblige. Again, you're going to receive a follow-up email with information from our sponsors probably tomorrow. So the sponsors have given us information about their respective clinical trials to share with you. Many of you have asked, how do I get involved? How do I learn more? So we have something coming to you that will explain a lot about the clinical trials that are ongoing in this space. So I would admonish you to read that information and then have conversations with your physicians about possibilities related to the clinical trials that currently exist. Not every clinical trial is going to be right for everyone. So you definitely want to have that conversation with your physician. And then we also have some resources within our CAR hub at lupus.org. So we have a lot of information, and that particular hub will be linked in the email that you receive in the days to come. In addition to that, the recording of the presentation tonight will be on our YouTube channel. So if you need to revisit, I know it's a lot to take in this evening. If you need to revisit this information, it will be available on our YouTube channel very soon. And it should be linked in the follow-up email as well. Again, if you have any questions, I'm more than happy to respond. Please reach out to me at buie@lupus.org. That's BUIE@lupus.org. Again, thank you so much to our incredible speakers this evening. Thank you to our donors, and we'll see you next time. Have a good evening.